Hi students, in the last class we have discussed our first chapter. We have just started the first chapter, the living world, and now we know that these are the basic characteristics of the living systems: growth, then reproduction, metabolism, cellular organization of the body, and consciousness. These are the basic characters of the living organisms, the living world. In this unit, we will learn more about the diverse forms of life. So if you want to learn, if you want to understand the diverse forms of life, we must know at least a basic idea regarding their classification. A basic knowledge of the classification of this biological system. So, in this section, I will introduce you one of the modern concept of biological classification. One of the modern concept of biological classification. And this concept is known as the three domains of life. This concept, the three domains of life, this concept was put forward by an American microbiologist. He was Carl Woese. He was an American microbiologist. So Carl Woese proposed, he put forward this concept of three domains of life. According to him, the three domains of life are the first one was Archaebacteria. The second one, Eubacteria. And Eukaryota. So according to him, he classified the entire organisms into three distinct domains, and these domains are above the kingdom level. And according to him, these three domains, these three groups have been evolved from a common ancestor cell. So they have a common ancestor. They have a common ancestor, and from this ancestor they have evolved. Okay, from this ancestor, the common ancestor, they have evolved. First domain, second domain, and third domain. So they enter organisms into three domains. These are Archaebacteria, Eubacteria, and Eukaryota. So these are the three domains according to whom Carl Woese. He was an American microbiologist. So Carl Woese, he classified the entire organisms into three domains. They are Archaebacteria, Eubacteria, and the Eukaryota. So now we must know about the basis of this classification. So what is the basis of this classification? So the first basis, the first factor was, it was the difference in one of the special organelle, that is the ribosome. You know ribosomes. Okay, the ribosomes are present in the cell, and those type of RNAs, the RNAs, these are present in the ribosome, is known as rRNA. Okay, rRNA. It's a special molecule because it is highly conservative, it is highly stable, it is highly protective in nature. Okay. So this molecule, it was one of the basis of this concept of three domains of life. The second one was the constituents of the cell membrane. Okay. So the constituents of cell membrane and this molecule, that is what ribosomal RNA. These were the basis of what this concept of, that what we call the three domains of life. Okay. So in the case of RNA, RNA, okay, it's a nucleic acid and it is formed of different units, that is known as what? Nucleotides. So there is a difference in the nucleotides. A difference in the nucleotide has an arrangement, a peculiar type of arrangement. That arrangement is known as the sequence. Okay, so difference in the nucleotide sequence of the rRNAs present in the cell. Okay, present in the cell. Okay, so why we are taking this rRNA? Because rRNA is highly protective in nature. They are highly protective in nature. They are highly conservative molecule, the most conservative molecule, and it is highly stable. Okay, and they have the similar function in the diverse living organisms. So the difference or similarities in the structure of rRNA can be taken as an evidence for what? Classification. Okay, so the difference in nucleotide sequence of rRNA present in the cell, it was one of the factor, one of the basis. The second one is what? The constituents of what? The cell membrane. The constituents of cell membrane. Okay, so these are the basics of what? This classification. Now we are going to learn about the basic characters of these three domains. So these are the three domains. Okay, Archaebacteria, Eubacteria and Eukaryota. These are the three domains under the classification of whom Carl Woese. Okay, and we know that the basics of this classification, it was what? The difference in sequence of rRNA, the difference in the structure or the arrangement of the nucleotides sequence of rRNA in their cells. And second one was the structure of cell membrane and its sensitivity to antibiotics, whether it is influenced by antibiotics. Okay, the sensitivity to antibiotics. So these are the two basics of this classification. So here Archaebacteria, Eubacteria and Eukaryota. So we are just going to describe this. Okay, so what are the basic features of these three domains? So the first one we can take the Archaebacteria. They are most primitive. They are the oldest organisms. They are very old. They are most primitive. Okay, and they are prokaryotic. They does not have a defined nucleus. The nuclear membrane is not defined. They are most primitive. They are prokaryotic. They have a unique rRNA. They are ribosomal RNA. They have a unique rRNA. their ribosomal RNA is entirely different from this eubacteria, and it has some differences with this eukaryota. So they have a unique rRNA, and they are resistant to antibiotics. They live in a diverse conditions, in extreme conditions. They can live in very extreme conditions of this nature. They are resistant to antibiotics. Their cell wall lack peptidoglycan. Their cell wall lack peptidoglycan. Then their cell membrane, it consists of branched hydrocarbons. Branched hydrocarbons. Their cell membrane consists of branched hydrocarbons. The main examples, the groups coming under this are what? Methanogens. They are methane producing bacteria. Methanogens or methane producing bacteria. Then halophytes. They live in extreme salt condition. You can say that the Dead Sea. You might have learned about the Dead Sea. So in the Dead Sea we can see this one. Halophytes. They are able to live in the extreme salt contained area. Then hyperthermophytes. Hyperthermophytes. They live in extreme hot condition. They are able to live in such a very extreme hot condition. So these are the basic characteristics of what? The group, the domain, Archaebacteria. Now here, eubacteria. They are also prokaryotic. They also have what? A unique rRNA. It is different from this one. That means the sequence of this rRNA is different from here. They are sensitive to antibiotics. Unlike this, they are sensitive to antibiotics. And their cell wall, it consists of what? Peptidoglycan. The cell wall consists of peptidoglycan. And their cell membrane has straight carbon chains. But here, just branched hydrocarbon chains. Here, what? Straight chains. There are examples: gram-positive bacteria, then gram-negative bacteria, cyanobacteria, and mycoplasma. These are some examples coming under what? This domain. Then here, last one. These are eukaryotes. Eukaryota. The cell design is what? Eukaryotic. They have well-defined nucleus and membrane-bound cell organelles. They have unique rRNA. So in each phylum, this domain, you can see that one unique rRNA, ribosomal RNA. They are resistant to traditional antibiotics. Okay, and they does not contain what? Peptidoglycan. So in this case, there are a diverse group of organisms, and in Animalia there is no cell wall. We know that in Animalia there is no cell wall, but in Plantae you can see the cell wall, and in them also there is no peptidoglycan. In Plantae, the cell wall is made up of cellulose, and they have straight carbon chains in their cell membrane. Okay, so these are the basic features. Here you can see the Protista, fungi, the Plantae, and the Animalia. So these are the groups coming under this eukaryotic domain. So these are the basic characteristics, okay, of these three domains. So now we have discussed the three domains and its basic characteristics. I think you might have understood this. Okay, so if you have any doubt, you may arise some doubt. There may be some difficulty. So if you find difficulty or any doubt, just ask me. You just inform me about this. Okay, and I will help you. Okay, if you have any doubt, you can call me, you can message me, so I will help you. Understand? In the next class, we will continue. Okay, so thank you all.